Okay, it's my very great pleasure to introduce Katrina Black, who started with me as a Europe student in her second year of university and stayed on to complete honours, and now she's reaching the end of her PhD. She had an absolutely stellar undergraduate degree funded by a slew of scholarships and awards, and so I've been privileged to have such an intelligent, motivated student who's been willing to take on a difficult project and put in the hard yards to see it through to completion. She's been a true asset to the lab. She's made a substantial contribution in terms of our ability to functionally assess the membrane-integrated proteins that we work on. So before she and David Miller, who supervised her up until the early last year, worked so hard at developing an assay, I was reliant on collaborators to carry out electrophysiological assessment. And actually, when I say reliant, I mean hamstrung. So ours isn't an easy channel for the electrophysiologists to work with, and Katrina's results sort of spurred them on to get their own experiments to work. So the fundamental research and the results she has produced are little short of spectacular in terms of what they reveal, and once they're published, they'll change the way that people think about potassium channels. So because of the implications, the bar to publication has proved excruciatingly high, and a year and a half on, we're still trying to find that magic control that will render the data incontrovertible, but we don't think it's too far off now. So Katrina's talk will focus on mechanism, but the consequences of her research are that ion channels, and in particular here potassium channels, the fundamental processes by which they switch on and off are neither totally conserved nor pore-centric. And it's really the first real handle, or we hope it will be, as to how to intervene effectively and discriminatively between the different classes. So I hope you'll enjoy what Katrina has to present in her seminar. I don't know what that did. That's telling me time, is it?
That was me, sorry.
Oh. Anyway, she's driven herself exceptionally hard to get where she is, and she's been a pleasure to supervise.
So thanks, Jackie, for that exceptionally kind introduction. And just before I start, I'd like to acknowledge the traditional owners of the land upon which I'm giving this seminar, the Wurundjeri people of the Kulin Nation, and I'd like to pay my respects to the elders past and present, as well as any elders of other Indigenous groups that may be here today. So the title of my seminar is Investigating the Mechanism of Gating in Kerr Channels. So I'm just going to start at a really basic level, which is, as you're, I'm sure, all aware, that the cell is surrounded by a lipid bilayer or a membrane. And this membrane is absolutely essential for separating the internal processes of the cell from the external environment. But the membrane is also a bit of a problem for the cell because the cell needs access to large molecules and charged molecules which can't diffuse through the membrane. So the cell needs these aqueous channels and pores in order to gain access to these polar and charged molecules. So one particular type of pore that you'll find in cells is ion channels. Now, ion channels are pore-forming membrane proteins that allow the movement of ions across the membrane. So ion channels are often selective, so particularly in the case of potassium channels, they only mediate the flux of potassium. They're very rapid, so movement of ions approaches the diffusion limit, and the movement is passive. So movement of ions is always occurring down concentration gradients and not in an active manner. So my PhD focused on a particular type of ion channel, which is the potassium channel, which obviously mediate the flow of potassium ions across the cell membrane. Potassium channels are very diverse, both in terms of their structure and their regulation. So this is a voltage-gated channel, which is controlled by changes in membrane potential, and this is an inwardly rectifying channel, which is controlled by the binding of ligands. And you can see that while they have similar overall protein folds, the minute details of their structures vary quite a lot. For example, the voltage-gated channel has 24 membrane helices, whereas an inwardly rectifying has only 8. So potassium channels play a number of very important physiological roles, ranging from very simple ones, such as controlling electrolyte balance and cell shape, to much more advanced ones. And this leads us to a discussion about why potassium channels might be interesting or important, particularly in a medical context. So if we think about some of the essential functions of potassium channels, one of the essential ones is setting a membrane potential, which then alters during action potentials. And so potassium channels are essential for normal electrical activity in the body. So this makes them essential for neural activity in the brain, setting our heartbeat, and allowing our muscles to contract. Potassium channels are also important in mediating signaling networks, and a really good example of this occurs in beta cells of the pancreas. So a change in membrane potential mediated by potassium channels is essential in the signaling pathway that ultimately results in the release of insulin. Potassium channels are also essential in overall electrolyte balance and filtering the blood, and so there are a number of types of potassium channels found in our kidneys. So these are all obviously essential functions for an organism. And if we have losses of function or we have mutations, we can get a number of different diseases. So in terms of neural activity, mutations can result in epilepsy or Parkinson's disease. In the heart, we can have a syndrome known as long QT syndrome, and mutations in channels can result in muscle ataxia. Children who are born with mutations in their pancreatic potassium channels tend to develop neonatal diabetes, and losses of function in the kidneys results in a renal disorder known as Barter's syndrome. So in my PhD, I was focusing on a particular family of potassium channels, and that is the Kerr family, or inwardly rectifying family. And I just want to take a bit of time to explain the basic structural features that will help you to appreciate the rest of my talk. So Kerr channels are tetramers. So here we have an overall picture of a Kerr channel with each of the subunits coloured in a particular colour. If we just take two of those subunits away and just look at it as two of the subunits, you can appreciate that they have a transmembrane region, so this is the bit that sits in the membrane, and a cytoplasmic region. The transmembrane region has two transmembrane helices per subunit, an outer helix and an inner helix. They also have a pore helix, which supports a loop region here, and this loop region is very important in mediating the selective nature of the channel. Now, the inner helices come together to form a region known as the bundle crossing. And the bundle crossing tends to have a large constricting residue, such as a tyrosine or a phenylalanine. And this residue tends to be oriented in such a way to produce a constricted pore, or a very narrow hole, at the base of the channel. And this is believed to mediate gating in the channel, or how the channel is able to stop the flow of ions. The cytoplasmic region has an immunoglobulin-like fold and is predominantly involved in regulating the channel state. So potassium as an ion has an energetic requirement for ligands. So if you dissolve some potassium ions in solution, you won't find potassium on its own. It will be surrounded by a hydrating shell of 6 to 8 water molecules. So this is fine in solution, but then what about as the ion needs to pass through the channel? Now, this region of the channel, the selectivity filter, is very, very narrow, too narrow to allow a fully hydrated potassium ion to pass through. And so if we take a look at the selectivity filter, we find that this requirement for ligands is instead met by the carbonyl groups from the backbone amino acid chain. So these carbonyl groups are very elegantly arranged such that they can perfectly coordinate a potassium ion. And this indeed is what mediates selectivity in this channel. Other ions, particularly sodium, which is also very, very common, have a different ionic radius, and it's smaller. So sodium cannot be coordinated in this way, and so cannot be accommodated by the selectivity filter. So this is just a little brief animation to show you how the selectivity filter works. So here we have the filter and 3 ions. You'll observe that the ions effectively jump between different coordination sites as they move through the filter, and this is how they maintain their chemical requirements they have for coordination. Now, in my title, I mentioned this term gating, and gating is a technical term we use in the field to describe the transition between a conductive channel and a non-conductive channel. And I want to describe the canonical view, or the view that is widely accepted in the field to describe how gating works. So here we have a non-conductive channel. In the non-conductive channel, we see the inner helices being in very close proximity to each other, producing a narrow pore through which a fully hydrated ion cannot move. If we have the binding of a gating stimulus, for example, a phospholipid, we see a conformational change, and the inner helices are further apart from each other, and this will allow a fully hydrated ion to pass through. So this animation is just to really tie these concepts together. Here we have fully hydrated potassium ions in solution outside the channel. They dehydrate as they enter the selectivity filter and rehydrate as they leave the selectivity filter. And this is how they're thought to then flow through the rest of the channel. So we have a ligand coming in to mediate gating, a conformational change. This is now very narrow, and the fully hydrated ion can no longer flow through. This is a non-conductive channel. The ligand is removed, the conformational change is undone, and now we return to a conductive state. So this is really to summarize the canonical view of how the majority of potassium channel structural biologists and physiologists believe channels to work. So if this is such an established view in the field, why is it a question that we've been asking in my PhD? And the reason that we're asking this question is because there still isn't direct structural evidence for this conformational change. And when I say that, what I mean is we haven't observed the same potassium channel in its wild-type state in both a wide and a narrow conformation. Now, the evidence that is often brought forth is to compare between structures of different families of potassium channels. So the first potassium channel structure which was solved is KCSA. And KCSA has a very narrow pore aperture with the inner helices close together, and so we see this narrow pore aperture. A later structure was then solved of a separate prokaryotic channel, MTHK. And in MTHK, the pore helices are much further apart from each other, and we see a wider pore aperture. Now, these are believed to be examples of a closed state and an open state. But it's also a valid interpretation to maybe think that these two states represent familial differences in potassium channels and not necessarily different gating states. I think this is further underlined by the fact that later structures have been solved of channels which are typically crystallized in a narrow pore, but they're also crystallized in the presence of activating ligands, which, if indeed this is the conducting state, it would make sense for an activating ligand to drive a structure to be in the open state. So how indeed can we test if a conformational change is occurring during gating? And the strategy that we came up with is to lock the channel in its narrow conformation and measure its function. If the canonical model is correct, we should observe non-functional channels because we are effectively stopping the channel from being able to access what is thought to be the active conformation. So here we have a little schematic of a potassium channel in its narrow or closed conformation. So what we can do is engineer cysteine residues into this region of the channel. And of course, cysteine is a very special amino acid because when you expose it to oxidizing conditions, you form a covalent crosslink. And forming a crosslink will mean that the helices will be connected to each other and will not be able to have the flexibility to move apart from each other as would be required in the canonical model. So of course, I did mention that the channel is a tetramer and not a dimer, and I don't want to confuse you by this diagram. So if we imagine that we're just looking underneath the channel here, what we're actually trying to do is crosslink each subunit to its neighbours on both sides in this sort of ring-based conformation crosslinking structure. We don't want to crosslink across the central conducting pathway. So the project itself has 3 stages. The first is a design stage. So how do we choose the right model channel and how do we make the right mutations? Then we have a structural phase, we do the crosslinking reactions, and then we assess how efficient the crosslinks have formed. And we can do that by crystallography, by mass spectrometry, and by SDS-PAGE. And then the final part of the project is functional. So having formed some crosslinks, we need to establish if the channels are functional still, and we can do that by at a population level and at a single molecule level. So to talk a little bit about the design stage, firstly, we want a channel with a narrow pore, which is very amenable to crosslinking. So if we just consider KCSA versus MTHK again, KCSA in its sort of low energy conformation that we observe in crystal structures has the inner helices close together. MTHK, on the other hand, the helices are very far apart. And so it's going to be much more easy to crosslink a channel in which it's happy to form inner helices which are close together than when they're far apart. So we want a channel which has a pore like KCSA. We also want a channel that we can make fairly good amounts of protein on fairly readily because we need quite a lot of protein to do structural studies. So this makes Kerr-Back 3.1 a good choice. Kerr-Back has a narrow pore aperture, so it should be easy enough to crosslink it. It's a prokaryotic channel, so we can make good quality protein from E. coli. And we already structurally characterized this protein in our laboratory, so we have a set of crystallography conditions. And I'm sure any crystallographer will tell you that having crystallography conditions makes any project much easier. So a second point that we want to consider is to remove background cysteines. So one of the things we want to avoid is artefactual crosslinks. We don't want the native cysteines that are found in the sequence of Kerback to spuriously crosslink with the engineered cysteines that we introduce. So what we want to do is remove the 3 native cysteines that we find in Kerback 3.1. So there are 2 in the pore region and 1 in the cytoplasmic region. And we simply just conservatively mutated these to valine and serines. So now we need to consider how we're going to make our mutations. So pretty obviously, it needs to be in the bundle crossing region. We also don't want to interfere with this central conducting pathway. So potassium needs a free pathway to move through. If there is a giant crosslinker in the way, then that's going to impede the movement of ions through the channel. Secondly is the position of this tyrosine residue, which is thought to mediate the constriction or the narrowness of the gate, which is in the canonical model is actually limiting the flow of ions. So if we remove this tyrosine or alter its position significantly, then of course under the canonical model you can interpret all results to mean that you've just altered the position of the constricting residue. So bearing all of this in mind, we have 2 mutations. One I'm going to call the 129-135, and one that I'm going to call the 133-136. So the 129-135 is shown here with little green markers to show where the mutations are, and here's the 133-136. The second orange one is just in this loop region here. So you can see the 129-135 is a little bit further up the turn of the alpha helix compared to the 133-136. And you can see that these are very close to the blocking residue of the tyrosine. So this is just to show you a model, so this isn't structural data, but to show you what we're sort of aiming for with these crosslinks. So if we just zoom underneath, it becomes a little easier to see. So this would be an inner helix and its neighbour, and we have a crosslink from position 129 to position 135, and likewise for the 133-136. Okay, so having decided on our mutations, we need to think about how we're going to crosslink them. And there are a couple of different ways that you can do this. The first one is this dibromobutane-mediated crosslink. So this is dibromobutane. You can undergo a nucleophilic substitution where we replace this bromine molecule with the thiol group from our cysteine, and we get this sort of very large, bulky crosslinker here. We can do an MTS1 MTS-mediated crosslink. So this is very similar to any MTS labeling that you may have performed, except we have a double-headed MTS with a single carbon molecule separating them. And then we get a longer, more flexible linker here. And of course, the obvious choice is a simple copper-catalyzed disulfide, which will produce the simplest, most direct crosslink. Okay, so putting all of that together, we have 6 experimental combinations. We have 2 mutations, and each mutation can be crosslinked in 3 ways. So I think to cast doubt on the model, the canonical model, we really only need strong evidence from one of these pathways. Any one of these pathways could show that a conformational change is unnecessary. But by doing it in 6 independent ways, I think our evidence becomes stronger because they will independently either verify or disagree with each other. So now that we've done our crosslinking, we can see how we've actually formed these crosslinks. Now, one of the most powerful methods that we have is crystallography. Now, crystallography essentially lets us directly observe the positions of atoms or electrons in molecules, and so it gives us the most direct way of observing whether crosslinks have formed and whether crosslinks have indeed formed at all 4 positions. So we obtain crystal structures of 2 of our conditions, the 129-135 in the presence of the MTS crosslinker, and the 133-136 in the presence of the dibromobutane. And I just want to briefly take you through one of these structures just so you can appreciate how the crosslinking worked. So here's the 133-136 cross-linked with the dibromobutane. The crosslinks are here, which is a little bit hard to appreciate the details of from this perspective. So if we just rotate our channel from the underneath perspective and zoom in, get rid of the cytoplasmic domain, I think it's a little bit easier to see how the crosslinking is work. So here's position 133, position 136 is in the loop region here, and you can see the crosslink, which is mediating this crosslink connection here. And we can see positions at all 4 positions around the structure of the channel. So if we just rearrange the helix here, you can get a look at one of these crosslinks mediating the interaction between these 2 helices. And the other point that I think is important to make here is it's very easy to just draw in whatever you like in a crystal structure. The important thing is how well it fits your electron density data. So this blue mesh is electron density data that we've collected from a diffraction experiment. And I think from this you can clearly see that there is electron density being between the two inner helices and a bulge of electron density where we would expect the dibromobutane to be. And so you can see that this density is all the way around in the whole ring, around the structure of the channel. And so we can be confident that there are crosslinks at all 4 positions that we need for the channel to be effectively locked in this conformation. Now, if we just rotate around a little bit again, I just want to highlight the position of this tyrosine 132. So again, this is thought to mediate the constriction of the gate. And so we don't want to alter its position. And I think you can see from this that tyrosine 132, despite the presence of these bulky crosslinkers and the mutations, it's still there doing what it would normally be doing. So this is the other structure, the 129135, crosslinked in the presence of the MTS. And I'm just going to take this through a little bit more quickly because it's essentially the same. If we just zoom in, this is the MTS-mediated crosslink here between position 129 and position 135. So again, we'll be able to overlay our electron density data, which is much nicer because this is a higher resolution structure. But again, you can really clearly see nice density reflecting the position of where the crosslinker would be. And this position, again, is in all 4 of the crosslinking positions. And again, the position of the tyrosine residue is unperturbed. So before we leave the crystallography behind, the other nice thing we can do is just take a step back and overlay several structures and compare how the overall fold of the channel appears. So here we've overlaid the wild type, which is in black, the reduced 133, 136 in red, and in green are the 2 crosslink variants. And I think you can appreciate this, how well these 4 mutations overlay with each other. And I think that it's quite obvious that the mutations and the crosslinks haven't significantly altered the way the channel folds or its overall structure. And the position of the tyrosine is unperturbed in all 4 conditions. So apart from crystallography, how else can we assess how efficiently we've formed crosslink? Now, in native conditions, both the uncrosslinked and the crosslinked tetramers will effectively behave the same. They're both tetramers of the same size. But when we introduce denaturing conditions, we will have a tetrameric version of the channel, which is covalently linked together, whereas the uncrosslinked channel will split into its separate monomers, which are smaller, and we can observe this difference. I just want to take a little time so we can think about the types of outcomes we could get from our crosslinking reaction. So this part of the table up here describes the different types of oligomeric states that we may see in denaturing conditions, and this is the efficiency of our crosslinking. So with no crosslinking whatsoever, we would observe 4 independent monomers. If we form 1 crosslink, we would observe a dimer and 2 monomers. If we were to form 2 crosslinks, they could either be neighbourly and form a trimer and a monomer, or they could be orthogonal, and we would form 2 dimers. Maybe we form 3 crosslinks, in which case we might see a trimer and an inwardly or internally reactive monomer, or we might see an incomplete tetramer where the last 2 cysteines do not react. And finally, we need to get to this point here. We need a tetramer in which all 4 crosslinks have formed, and so we're able to say that we've locked the channel together. So SDS-PAGE is a technique which would allow us to differentiate between the tetramers and the other oligomeric states. And SDS-PAGE is also very useful because we can use it as a screening technique to make sure that in our functional experiments, we're only choosing the most efficiently cross-linked samples. So here we have an SDS-PAGE gel. This is just done in detergent, in DDM. And I've chosen this gel not because it's the best example of cross-linking that we have, but because it clearly shows how we can differentiate between monomers and tetramers. So at the top of the gel, we have tetramers which are resistant to being denatured by SDS. So you can see in our cross-link conditions, that's by far the predominant species that we have. And then at the base of the gel, we do have trace amounts of monomer. And we compare that to the reduced mutants, you can clearly see that's predominantly running as a monomer. So we can effectively see how efficient our cross-linking is. This is a gel of actual results from a functional assay. So these are the sorts of results that we've actually chosen for our functional assays, and this was performed in liposomes. So I think the first thing you can take from this is that we've been quite selective and you can't directly observe any monomer in these populations. And the second thing, which is quite obvious, is there's quite a lot of variability in the efficiency of how much protein we're getting in our liposomes. So the same amount of protein was added to these liposomes, and the same amount is run on the gel, but you can see much more protein in this lane and this lane compared to this lane. So that's just something to bear in mind as we move forward. So another technique we can use to try and establish how efficiently we've cross-linked is mass spectrometry. Now, mass spectrometry is a very sensitive technique, and it can differentiate between both the tetramer populations and the other oligomeric states. And it can also differentiate between our two tetramer populations, which makes it very, very useful. So firstly, I just want to go through how mass spectrometry can allow us to differentiate between the different oligomeric states. So here we have a mass spectra. This is our mass to charge ratio, and this is the relative proportion of a particular species. Now, this is a native mass spectrometry experiment. So this is not protein which is being tryptically digested and allowed to run in the mass spectrometer. This is a whole protein flying in the mass spectrometer. And these experiments were performed by perhaps the world expert in this technique, which is Carol Robinson and her postdoc Yanni Bowler at the University of Oxford. So here we have the wild type, and we have two peaks, one here and one here. Now, this peak here represents the oligomer. This is a larger species, and so this represents the tetramer. This is a much smaller species, and so this peak represents the monomer. And so we're seeing at least partial movement from the tetramer into the monomer state. Now, we can compare this with the 133-136, which has been cross-linked in the presence of dibromobutane. And then you can see that we don't really see this other peak. We only see the predominant species here, which is the cross-linked tetramer species. And so from mass spectrometry, we can really clearly see that we have indeed formed oligomers and not, we don't have a set of uncrosslinked tetramers. Now, the other thing that mass spectrometry is very useful for is differentiating between partially cross-linked and completely cross-linked tetramers. And the way that you can do this is by careful analysis of the masses that you would see in a mass spectrometry experiment. So here we have our uncrosslinked tetramer, which has a mass of 134,218 daltons. Now, we have a fully cross-linked tetramer, and the exact mass that we see will depend a lot on what we use to cross-link it. But let's say we use a dibromobutane. It's very big, it's very bulky, it's very easy to see masses using this cross-linker. So a fully cross-linked tetramer will result in an increased mass over this sort of baseline mass of 752 daltons. Then we have our incompletely cross-linked tetramer. So if we have an incompletely cross-linked tetramer, what sort of masses might we see? So firstly, we might just have no reactions occurring at this last cross-linking site. Then we would expect to see a mass increase of 565 daltons. Let's say only one of the cysteines react. That will produce a mass difference of 834 daltons. And maybe each cysteine independently reacts with a dibromobutane, and so there's not a free cysteine left to mediate that last crosslink. And that will produce a mass difference of 1,102 daltons. So we can analyze our data carefully in all of our cases and compare the masses that we see in the tetramer compared to the non-cross-linked variant and observe what that mass difference is. So this is our calculated masses for our various mutations. So for example, the wild type is 33,606 as a monomer. The 129-135, when it's reduced, is 33,554 daltons. We can multiply that by 4 to get the tetrameric size. And then we can run our mass spectrometry experiment and observe what masses we see. So I just want to focus in on the 129-135 butane because I think that really exemplifies how this works. So in our mass spectrometry experiment, we observe a mass of 134,962 daltons. Now, if that had no crosslinks, we would expect a mass of 134,218 daltons. The mass difference is 744.5 daltons. Now, we know that the calculated mass difference of a perfectly cross-linked tetramer is 752, and they're pretty damn good matches there. So, and we can do the same experiment for the MTS and the 133-136, and you can see in all 4 cases the mass differences that we observe in our mass spectrometry experiments always fit really nicely with what we would expect for 4 complete tetramer crosslinks. It's a little hard to do the experiment for disulfides because the difference in mass is only 2 protons, and so that's going to be not really figure outable within the error that we see in the experiment. Okay, so how wide? So if we have indeed formed these crosslinks, how wide is the pore aperture in these crosslink mutants? So I think the dibromobutane molecule is again a really good example to think about what sort of pore aperture size that we're seeing. So here's our dibromobutane molecule cross-linked protein, and you can see the dibromobutane cross-linker here. Now, dibromobutane is a cyclic molecule, so the bonds that it has are very rigid, and there's going to be very limited rotational and conformational flexibility in its bonds. And so it becomes quite hard to imagine how you would get much stretching of these bonds to accommodate changes in the location of these inner helices. So it's reasonable to surmise that we're limiting the pore aperture here to around the size of this intermolecular distance, which is around 4 angstroms. Now, in comparison, a fully hydrated potassium ion has a size of around 8 angstroms in diameter. So this pore aperture should still be too small for a fully hydrated ion to pass through. Which leads us nicely into our functional experiments. Do our crosslink channels conduct? So the first way we can go about answering this is to do a fluorimetric liposome assay to assess function at a whole population level. So here we have a liposome. Here is our potassium channel. Here is potassium ions contained within the vesicle. And here is a proton-sensitive fluorophore called ACMA. So this will be at maximum fluorescence at the beginning of the experiment. To begin the experiment, we add a protonophore called CCCP. When CCCP is present, protons are able to move into the liposome freely. Protons moving in will negate the buildup of negative charge, which will have occurred inside the liposome, that allows free egress of potassium ions out of the liposome. And this will result in a protonation of the ACMA, and this will result in a decrease in fluorescence. At the end of the assay, we add a potassium-specific ionophore called valinomycin. So when valinomycin is present, it essentially allows free movement of potassium ions across any membrane that occurs in the assay. So this will allow potassium to move out and reach its final electrochemical equilibrium, or like 100% flux point. At this point, ACMA will be at its minimal fluorescence level. So this is an example of the type of results we see in this assay. So in blue here, we have results when we have no channel present. So this is effectively like empty liposomes. So you see a small change in fluorescence, which is probably mediated by leakage of protons. But when we add in a wild-type channel, you can see a much more nice decrease in fluorescence over time here. So the decrease that we see from this point to this point is the fluorescence due to potassium flux through the channels. The drop that you see here is the fluorescence drop due to valinomycin. As a negative control, we could add in a spermine block. So spermine is a polyamine, which is able to move up into the conducting pathway of the channel and stop the flow of ions. So when we have spermine present, we don't see the change in fluorescence anymore, and we can be relatively certain that we're working with a potassium channel here. And then we can also use our crosslink variants and see that they're functional as well. So this slide summates all of our functional results that we've obtained. So the results are the mean and the standard error of the mean for 3 independent experiments. And each independent experiment was performed in triplicate as technical replicates. So you can see when you have liposomes only, we do have a small change in fluorescence of around 10%. But when we add in wild-type channels, the fluorescence changes between 30 to 40% in both the wild-type and our cysteineless background channel. Now, we've got a few control experiments here. Here is a sodium chloride control. So it's important to establish that we are working with a potassium-specific channel and that we haven't accidentally pulled through a non-specific porin from E. coli, for example. And so by using sodium instead of potassium as the ion, which doesn't mediate any flux, we can be more confident that we're working with a potassium channel. In order to further sort of control for this, we did an empty plasmid control where we expressed from a plasmid not containing our gene for our potassium channel and treated the samples in exactly the same way. And when we do this, again, we don't see any change in fluorescence. So it's not likely that there's an E. coli contaminant interfering with our results. And this is our spermine block experiments. And spermine is relatively selective for potassium in its blocking ability. So it gives us some more confidence that we are, in fact, measuring flow of ions through a potassium channel. So here are our results for our non-crosslinked channel, the 129 and the 135, which are just as functional as the wild type. And in all 6 of our experimental conditions, both mutations and all 3 cross-linking conditions, we can clearly see that the channel is still functioning. So this is just gels summarizing the actual the actual channels which were used in these experiments. So you can see, with the exception of this one MTS mediated case where there clearly is monomer, we can't observe any amounts of monomer in any of our cross-linked states. So I think we can be fairly confident that there isn't large populations of uncrosslinked channels mediating these functional results. So the other functional method that we tend to use is called electrophysiology, and that allows you to do single channel recordings. So to do this, we use something called painted bilayer electrophysiology. So in painted bilayer electrophysiology, we have two bathing solutions, a cis and a trans. And separating the cis and trans baths is this bilayer that you sort of paint on. In the cis bath, we have high levels of potassium. In the trans bath, we have low levels of potassium. And in each bath, we have an electrode. So what happens is we can take our liposomes, the same liposomes that we use in our other functional experiments, and fuse them with this bilayer. A channel will spontaneously insert here, and an ion will be freely able to move across. And we can measure this as a small change in current using these electrodes. So this is a typical sort of result that you would get from an electrophysiology experiment. So you see when the line is at the top here, that means the channel is not functional, it's closed, and we're not seeing any change in current. When we see results down here, that means that we're observing a current, and that's mediated by the channel. That means the channel is conductive. So I think the other thing that you should appreciate from this is, firstly, the results are digital, so you either have on or off. You don't have intermediate channel conductances. And secondly, it's very common for channels to sort of flicker between an on and an off state. So of course, we did these experiments for our channels. So here's our cysteineless background, which is open predominantly, but occasionally flickers into the closed state. Oh, and I should say that these experiments were performed by Derek Laver and Paul Johnson at the University of Newcastle. We can add in our spermine blocker, which decreases the conductance very significantly due to it blocking the ability of ions to move through the channel. We have our 133, 136, and 129, 135, which haven't been cross-linked, and again, they're predominantly open, predominantly conducting. And this is also what we observed in all 6 of our cross-linked cases, the dibromobimane, the MTS, and the disulfide. And the bimane seems to be pretty happy to be gating. It opens and closes pretty regularly. So the final question that we might want to consider with these functional results is it possible that all our results are being mediated by a small population of uncrosslinked channel? So we'll do a little thought experiment here. If the canonical model is correct, then indeed all of our functional results have to be due to a small population of uncrosslinked sample. And I want to note that we don't actually be able to see these in any of our gels. So there should be a relationship between the amount of protein we put in and the fluorescence change that we observe. And there should also be a limit to the sensitivity of this assay. There should be a minimal amount of protein that we can put in to see a fluorescence change that is statistically different from liposomes with no channels. So can we establish this threshold or this relationship? And so can we determine if a very, very small amount of protein, i.e. the uncrosslinked channels that may exist in our samples, could result in the fluorescence change that we've observed? So here what we've done is a proportional reconstitution. So we have here, like this represents 100%, and 100% is the amount of protein that we would typically include in our normal functional results. And then so this is representing adding 2/3 of that amount of protein, 1/3 of that amount of protein, 10%, or adding no protein. And you can see we do have a nice relationship. Increasing the amount of protein increases our fluorescence change with a maximum of adding 2/3 of the normal amount of protein, and then it dips a little bit at the end. And you can see this reflected in this SDS-PAGE gel. More protein present, more fluorescence readout. We did the same thing with the 133, 136 disulfide, and here you can see that again, a similar relationship, more protein, more fluorescence readout, with a little dip at the end, and this is reflected with more protein, the maximum amount of protein, where we get the maximum fluorescence. This relationship doesn't really seem to hold in between these two samples, so obviously we seem to have any more protein in the reduced samples than the crosslink to get the same fluorescent readout. But I think what you should take away from this is that you seem to need quite a lot of reduced sample to get a fluorescence change of around 40%. Even a 10% change in sample produces a fluorescence change which is different, which is much closer to 20%, and these are different from each other. So I think it becomes very easy to say now that a sample which has so little protein that you can't even observe it on SDS-PAGE must have a fluorescence change which is very low, very close to or indistinguishable from having no channels present. And it seems very difficult to see how such a population could result in a fluorescence change of around 40% that we observe. So that brings me to my conclusion, which is a conformational change in the bundle crossing region is not required for normal conduction and gating in Kerr channels. Now, so that sort of leads us to a question: how do we explain these results? If it's true that the pore aperture is very narrow, around 4 angstroms, and a fully hydrated potassium ion is 8 angstroms, how do we reconcile these facts with the results that we see? So the first thing I just want to say is that we can actually observe potassium ions sneaking past this tyrosine residue in our crystallography experiments. So it is something that we can see in our experiments. Secondly, the selectivity filter is also a very narrow region of the channel, and we know how ions get through this part of the channel. They lose their hydration shell, and they move through as naked ions and are coordinated instead by these carbonyl groups. So what we propose as a model to explain our results is to think of the channel as having separate zones through which the ions transit. At the top of the channel, we have a fully hydrated potassium ion surrounded by its water shell. In the selectivity filter, it's a naked ion, and it's instead coordinated by the carbonyl groups. And then, as it moves out of the selectivity filter, it regains its coordination shell of waters. Now, when it reaches the selectivity filter, what we see is what we're proposing to happen is that we have a transitional water shell. So the ion is passing through here, it has a group of water molecules, but as it gets to this region that's narrow, it loses some of those water molecules and then gains some very quickly on the other side of this very, very narrow pore aperture. And so it's able to have a transitional water shell, which it doesn't. The water shell it has at the bottom here is just different from the one that it has at the top. And you can sort of think of this as like a transitional state in chemistry in which bonds are being broken and made in a very rapid period of time. Remembering ions are going through this very rapidly, approaching the diffusion limit. And so this is kind of how you could explain how a single ion is able to move through this narrow pore aperture. And this little animation is just sort of to establish this. So we have ions that are dehydrated as they exit the rehydrated as they exit the selectivity filter, and then as they get to the bundle crossing region, they lose some waters and gain some waters as they pass through the constriction, and effectively move through without their hydration shell. So my acknowledgements, of course, I need to acknowledge my supervisors, Jackie and Dave, and now Peter Coleman, who have taught me everything that I know about science, and this project, of course, could never have happened without them. The other members of my lab, Xiao Wen and Agalia, who have been really good lab mates and I've really enjoyed working with, and have been exceptionally helpful in terms of experiments, as well as just chats and helping to think through problems. Ollie, who was the previous PhD student in the lab and was involved in working out some of this project and doing some of the early structural work. Chris Burns, who made some of the reagents that we're using. My committee, Melissa and Francis, who have been very supportive, and I thank them for being on my committee. Structural biology, which is an amazing division to do a PhD in. It's been very supportive, and I really couldn't imagine doing a PhD in a better division than structural. Yanni and Carol at the University of Oxford, who kindly did the mass spectrometry experiments. Paul and Derek at the University of Newcastle, that performed the electrophysiology experiments, as well as Monique and Adam and Jamie at Victor Chang, who also performed involved some electrophysiology. Brian and Andrew did some molecular modelling for us. Tom at the synchrotron helped us with diffraction. Janet at CSIRO helped with crystallography. My family and friends, who've been very supportive through some of the tumultuous times in the PhD, and funding from my APA and the Edith Moffat.
Great. I think just before I ask for questions, I'll apologise for my remissness earlier in not acknowledging the traditional owners and custodians of the land. There was no disrespect intended. Now, Katrina will take questions.
Robin?
I've got a couple of questions. First one, you only showed 2 crystal structures for your 6 different iterations. Was there a reason for why the other ones didn't crystallize?
So they did crystallize, but they didn't fract well. So we couldn't obtain good enough diffraction data to actually obtain structures. So yeah, that's why.
And the second question is, in terms of human potassium channels, how varied is the width for?
So it depends a lot on the family of the channel. So some channels have wider pores and some channels have narrower pores. Inwardly rectifying channels always have a narrow pore. And so I think it's pretty clear that there is variation in the width of the pore, and we can directly observe that. The question is, the width of the pore, is it something that varies within the channel? Is it a conformational change? Or do different channels have just different pore widths, and that is reflected, for instance, in increased conductance or different regulation? Davo?
What's the mysterious control that you've been asked to produce, and why do they want to see it?
Well, basically the reviewers say that our functional results...
Mysterious control. Where did he come up with?
Because you said that we're waiting for more control to get our structure published, right? So I think the reviewers tend to be very disbelieving of our results. So they seem to think that it's impossible for us to establish that there is no population of uncrosslinked channels, and so we can't say for sure that we're not seeing function from just channels that are either incompletely cross-linked or not cross-linked at all, and that's mediating all our functional results. So we've spent a lot of time trying to shore up, doing extra controls. I mean, some reviewers have suggested that the entire thing is done through it. There's a contaminant in there that's resulting, that we've done extra controls to establish there isn't a contaminant. So I think because it is such a controversial finding, we need to have such a well-controlled set of experiments to get over the skepticism that exists.
And is the chief controversy the hydrated ions come through and bring the water with them? Is that the...
Yeah, so so a lot of this original model came from very early experiments. So potassium channels were discovered very early. As soon as we appreciated that action potentials were a thing, there were people doing experiments in the 40s on squid axons and knowing that potassium was moving across the membrane. So it wasn't until the 90s that we had any structures of these channels. And so there were a lot of early experiments on accessibility. So we could see that in different states of function, sometimes you could get a bulky molecule inside the molecule and sometimes you can't. And so that was interpreted to mean that there must be a steric gate that was either allowing that molecule in or not allowing that molecule in. But if you actually go back to a lot of those results, a lot of the residues they believe were involved in those experiments aren't actually inside the pore or wouldn't really make sense to be mediating a steric block. So I think this is one of those areas in science where a model has been proposed very early by some very eminent figures in the field, and it just hasn't really been directly tested. And I think that makes people sort of accepted it without really critically thinking about it. Eddie?
Very committed for my own life as well. But I was going to ask, so if it's not a conformational change which regulates the on-off state of the gate, then what does?
So that's something that's obviously a very speculative answer. So we don't have any direct evidence either way. But I think what you can sort of pick up from this is that hydration is really important for potassium ions. So what if instead of a steric gate, it was controlling the accessibility of ions to water molecules themselves, so whether there was actually able to get that group of waters at the base of the selectivity filter present, which was enabling them to either be on or off. So if there were little trajectories for water to get in and you could block that in some way, then you would turn the channel off, and if you could open them up, you could turn the channel on. But that's very speculative.
Well, I guess it could be to do with overcoming the ability of hydration.
Yeah. I think people have thought about it in a very physical way and not in a very chemical way, and that's kind of where I think the gating mechanism may actually be. Raf?
Can you see the ion stabilization, that second step? You know how the carbonyl backbone stabilizes the dehydrated potassium with selectivity filter.
Yeah.
Can you find anything for the second gauge?
So we can observe this. So we can observe some stability of the ions here in our crystal structures, but they're not as well defined as they are in the selectivity filter, no.
What would happen if you put something positively charged or negatively charged in there? Can you play with conductance in that way?
Yeah, so some people have actually done those sorts of experiments. So there's so KCSA is one of the big channels that people do mutagenesis on, and there was someone who basically removed all the charges in that region, or to produce only negative charges in that region. And that actually produced like a basically non-functional channel, so it completely destroyed the normal conformation in that area. So it seems like mutations that you make in that area are important for the function, but wouldn't simply just result in an open pore. Melissa?
So in that selectivity filter, you've got all those really nice carbonyl groups that
Yeah.
are coordinating the potassium as it goes through, but there's nothing similar with the base.
Yeah, so I think
What's coordinating the waters?
so. I think that it's not a very long, it's not a very big jump that the iron has to make to get through one residue, right? So it could have a set of waters here and a set of waters here, and it can access both of them. Selectivity filter is much longer, so you need to have intermediate coordination sites. So the iron basically jumps from here to here and out. Like, it doesn't go through every coordination site. So it's easy to imagine that it could go from here to here by just jumping from water to water. Matt?
Did you try any cysteine combinations that didn't work, or did you just have an incredibly good eye?
No, we tried other cysteine combinations. Some of them didn't produce very good crosslinks, so the crosslinking was just very inefficient, and so these were the two that produced the most efficient crosslinking. Yep.
It's a great talk.
Thanks.
I don't know much about ion channels, so I was just wondering about what gives the directional force of the movement of the potassium ions. Is it like a stochastic process where some go back out, or do they all go in one direction just through that carbonyl tunnel and through the inside?
So the direction is actually kind of opposite to the way I've shown. So most of the time, potassium is coming from the inside of the cell to the outside of the cell because that's the diffusion gradient. But in inwardly rectifying channels, there's actually, so they're called inwardly rectifying because there's a rectification effect. So polyamines and divalent ions actually are able to move up into the channel and stop flow from going the other direction. So there's different mechanisms that occur in different types of channels. Yeah?
And how constrained are those the tyrosine groups, those proteins at the very end? Are they quite locked in, or can they kind of rotate?
I think they can rotate fairly flexibly. So
Would it be enough for the ion to go through?
no, I don't think so, because there's still not really enough space, even if you move them all. And you have to remember, they're not really going to want to be too close to each other. So there still wouldn't be 8 angstroms of space inside that pore. Peter?
So if your idea about restricting access of water is correct, that would mean that there wouldn't be any free water within that cavity. Because otherwise that could move around and act as the solvation desolvation. So is that what you see in any high resolution structures? Is all water always coordinated with the potassium?
No, I mean, there's definitely free waters available in the structure, but I also don't know if we've captured a fully closed state in the crystal structures either. So unless you can correlate between the two, it's very hard to know what you're looking at. So yeah, I agree that, obviously you can't have a huge amount of free water in there, but for every potassium molecule, you need quite 7 or 8 molecules of water. So it wouldn't be hard for several potassiums to suck up quite a lot of water in there.
So how many potassiums do you ever see? What's the most number of potassiums you see in there?
In a crystal structure?
Or are they just not coordinated?
They're not always ordered or coordinated. So really the selectivity filter ones are the most coordinated, and then you need to go to quite a low sigma level to actually see them near the tyrosine residues.
So have you looked at how many you could fit in there?
No, I haven't looked at that. Angus?
I can't remember exactly. Are the tyrosine hydroxyls in a sort of arrangement where they could desolvate the potassium ion, or the?
I mean, that's potentially true, but in other channels it's a phenylalanine or an isoleucine, so it can't really be a universal mechanism if it's not always a conserved tyrosine, I think. Jason?
So if you put this model onto all the other structures of channels, so I don't know how many potassium channels there are, does everything make sense? Is it, do you know what I mean? You're mentioning there's different residues at that gating position.
So I think that actually part of the problem in the field is that there is assumed to be a universal way that channels gate. But we have all this extra diversity, they barely look very alike to each other. So I don't think necessarily that we're claiming to have one model which describes gating in every channel. I think this is pretty good evidence for inwardly rectifying channels. But I don't think that it could definitely be possible that there are different gating mechanisms in different channels, which actually be very exciting because that means different mechanisms for controlling them with drugs, which will be specific to a particular type of channel.
It's more of a rationale for what she's seeing than a model, just to clarify. Yeah.
Tony?
So as stripped ion migrates through there, the free energy has to drop at some point.
Yeah.
And is ion, or have you considered the molecular dynamic free energy calculations to give you some support that that's the most likely way of the ion
Brian.
transporting itself?
Yeah, so Brian has done some calculations for us, and he's still in the process of really confirming that they're correct. But it was looking like that there wouldn't be significant energy barriers to this as a proposed model.
If I can add something in, I can't see where Tony is.
He's just here.
Oh yeah, but there is actually captive water in cavities that's positioned really by the structure of the Kerr pore. So it's not that it would ever be naked going through. Yeah. Okay.
Rich?
Sorry if I missed it, but how many copies do you see in the asymmetric unit?
So it depends on the structure. So some of them there's one, and some of them there's two.
Yeah, there's two.
Yeah. So it depends on the space group. So in some of them there's fourfold symmetry, and some of them there's not.
Does that change with the different crosslinkers?
Yeah, it has changed with different crosslinkers, but it also we see different space groups in the wild type anyway. So it doesn't really seem to be correlated with the crosslinkers.
I think. Right.
Melissa, last one.
Last
I
one.
just wanted to ask, so the assay that you did diluting the protein with the uncrosslinked.
Yeah, the proportional one. Yeah.
You needed a lot more protein with the uncrosslinked one to get
Yeah.
the same amount of lipids.
Yeah.
Why do you think that is? Do you think that there's just some that's not tetramerizing in the sample? Why?
So that's a good question. I did actually prepare a slide for that because I thought someone would ask. So the first thing that maybe there's a contaminant in there that's like if there's a porin in there and it's the same amount in all of them, yeah, then you're going to see that. But I think we've established that that's not the case because of the extra controls that we've done. Secondly, maybe the crosslink channels are more functional for some reason. Like maybe they're open more often or have a higher probability of being open, and then you don't need as much protein to get the same result. Thirdly, you have to think of this as a ratio of protein to liposomes. So yes, there's more protein, but are there the same amount of liposomes? So maybe in the reduced samples, there's more protein, but also more liposomes, and maybe in the crosslink samples, we're losing more protein, but also losing more liposomes, and the ratio is the same. So I don't know the answer yet, and I have to work out that in the next little bit.
I guess the fact that your highest amount of protein was in the 2/3 sample suggests that the reconstitution must be negatively, or must be inhibited when you have too much protein.
Yeah, I think it's a precipitation issue. I think if you have just too much protein, it starts to precipitate and drag the lipid out with it, and that's why you get a reduction in your readout. Yeah.
Right. Oh, Peter wants. So
Okay.
This is the very last one.
So why is the flickering opening and closing rate so different in the case of the cross-linked channel?
I absolutely have no idea. Do you know? I mean, there's no reason to think that the bimane crosslink, which is the most flickery than the others, is like it looks the same structurally. The crosslink is probably more rigid than the MTS, which you'd think would allow a little bit more flickering to occur.
That particular structure is a little less stable because when the bromobimane binds, you do get a slight distortion of the cytoplasmic domains, not the pore. Now that might correlate with the stability.
So do any of the crosslinks show a flickering rate that's like wild type?
That's good. So I only have a wild type cyslus in here. So
I think they're not actually the final traces that you have there. But
I mean, I don't necessarily.
Before that, you had the mostly closed and flickering open, right?
Yeah, but this is not necessarily. It's just an example trace. It's not of a Kerr channel. It's of a different ion channel. It's just to explain how you can interpret the results, really. Yeah.
And then I've got one objection. Go
Yep.
to the cartoon, the other cartoon. So I wouldn't want anyone to leave here thinking that there was a mirror. No, not that one. Well, that one's almost. There was another one. Keep going.
That one?
That one. Don't anyone think there's a mirror of symmetry?
no.
It was a lot easier to draw it with that. Yeah.
Okay, I'd like to close it there, and please feel free to join us for drinks and biscuits, and please help me thank Katrina again for that.